So as you said, in 2017, I opened my new lab here in Israel in the Hebrew University. And we're a theory group interested in different aspects of theory of evolution of plants, as well as general theoretical questions. And we currently have 3 main lines of research in the lab. So you actually mentioned 2 of them already. So the first one goes back to my projects in IST, where I was thinking about the evolution of networks and how genes that function in a network actually can constrain and affect each other's evolution. So then I was working about gene regulation, and now I'm asking these kind of questions in the context of self-incompatibility in plants. Second project is a collaboration with a colleague here, where we try to fit a computational model that will predict how flowering of olive trees depends on winter temperatures based on data that my colleague is collecting. And the third one is evolution on high-dimensional fitness landscapes, and this is the project that I'll be talking about today. So I will start with a brief introduction about fitness landscapes so that everybody here is on the same page, and apologies for people that are already familiar with the basic things. So the concept of fitness landscapes goes back to Sewell Wright in the 1930s, who offered to look at the mapping between genotypes to their fitness at some sort of a landscape like the one that we see here, where the metaphor goes like that. So a location in the plane, the XY plane, represents a genotype. Every location is a different genotype, and the height represents the fitness, like the hill here or the valley that we have here. So this is just an upper view of the same topographic map. So importantly, this is not just a mapping from genotype to fitness, but actually this also includes a measure of distance between genotypes. So genotypes that are far away from each other in this landscape are also far away in terms of the number of mutations needed to go from one genotype into the other. So this metaphor can also help us think about the evolutionary trajectories that a genotype can go through. Okay, so this was fine to get some intuition, but actually the former picture is a bit misleading in at least 2 aspects. So one of them is that the fitness landscape is not 2-dimensional. Its dimension is actually much, much higher, depending on which genotype exactly we study. And also it is not continuous, it is actually discrete. So instead of this 2-dimensional topographic map, we should think about a graph in which we have discrete genotypes. Every genotype here is a node in the graph, and the edges between these nodes connect genotypes that differ only in one position, namely a single point mutation can take us from one of them to the other. And the dimension of this graph now depends on the length of the sequence that we are interested in. So I'm showing here a simple example in which we have a length for genotype. In this case, these are all the combinations of 4 antibiotic resistance mutations in a particular bacterium, Methylbacterium extorquens. 1 stands for there is a mutation in that position, 0, there is no mutation. So we have a total here of 16 different genotypes, and now we can go from no mutation at all to 1 mutation in the first place, the second, third, or fourth. The color here designates the fitness. The darker is the fitter, where in this case, fitness is the relative resistance to antibiotics. One more thing to notice is that the direction of the arrows here shows the direction in which fitness increases from low fitness to high fitness, namely the direction that we expect evolution, natural selection to take the population. So I marked here the two genotypes that are local maxima in this landscape. If you look closely, then all four arrows enter into these genotypes, and there is no arrow leaving. Namely, all single mutants are less fit than this genotype, but also than this one. Okay, so now this also brings us to the question of what would be the evolutionary trajectories that we should expect, and whether the population will end up being in this maximum or perhaps in the other one. So this was only a simple example where the dimension of the landscape is 4, but it can get much higher than that. This is an example from Payne and Wagner that created this fitness landscape of the binding affinity to a particular mouse transcription factor, and the fitness here, the phenotype, which is the fitness, is the binding affinity of all DNA sequences of length 8. So here we have a cloud of a much larger number of genotypes, and it's much harder to get an intuition what is going on here and what are the evolutionary trajectories here. So the recent advances in experimental techniques, and in particular in mass sequences, enabled now the measurement of larger and larger empirical fitness landscapes, going way beyond the 16 different genotypes that I've shown you. So we see in the recent years experiments that reach tens of thousands, or getting even close to 10 to the 5th different genotypes. These are only a few examples, and there are more of them. So this one is the green fluorescent protein from Kondrashov's lab. And this is the snow RNA here in yeast, and so on. And we're talking here about 50 to 60,000 different genotypes that were measured. So another important difference here is that the earlier measurements often took mutations that were found naturally and then measured all their combinations. So then the choice of the genotypes that were measured was biased in that sense. And these more modern experiments very often use random mutagenesis, so perhaps they probe the fitness landscape in a less biased way. Okay, so fitness landscapes are huge, but let's get some intuition. Where are the modern measurements of tens of thousands of genotypes with respect to the actual size of fitness landscapes? So if we think of a typical protein of, let's say, a size 300 amino acids, then there are a total of 20 to the power of 300 different possibilities for such a protein, simply enumerating the number of the peptides of that length. So of course, not all of them would be relevant proteins. Some of them may not fold or be unstable, but still we're talking about very large numbers. And this is just for a single protein, and the organisms, of course, have more than one protein. So just for comparison about the size of these numbers, then the numbers of atoms in the whole universe is estimated to be 10 to the 78 to 10 to the 82, which is much smaller than this number that I have here. Now, the state-of-the-art experiments checking 10 to the 4 to 10 to the 5th different genotypes suddenly don't look that great compared to such large numbers. But does biology do better? So a typical microorganism population size is 10 to the 7 to 10 to the 9. So that is larger than the number of different genotypes measured in such experiments. And of course, populations have lived for a large number of generations. So they could have explored in that time many different genotypes. But then it is not so sure that even biological populations have explored the huge number of possible genotypes that there is there. So that's the bottom line here, is that these measurements, as comprehensive as they are, then are only scratching the surface and are actually only measuring just tiny bits, or the tip of the iceberg of the real fitness landscapes. Okay, so I was talking until now about the mapping and how many genotypes are mapped in the experiments. But the interesting question that we can use fitness landscapes to ask is what are the evolutionary trajectories that we should expect? So let's go one step back and just recall what the basic one locus models of population genetics tell us. So these models assert that given enough time, then we should expect a mutation-selection balance between the fittest genotypes and deleterious mutants of this genotype. And if Q is the proportion of the mutants and 1 minus Q of the fittest genotype, then the relative proportion of the deleterious mutants simply depends on the ratio between the mutation rate mu and the selection against those mutants S. And if we think about the low mu over S, the strong selection weak mutation regime, then most of the population is expected to occupy the fittest genotype, and only a very small proportion will be the deleterious mutants. If mu over S is higher, then we should expect a larger proportion of the deleterious mutants, but still there should be some balance between fittest and less fit. So the major thing that is lacking in these models is that they do not have any landscape structure here. So they actually assume that all mutations are equally accessible, and in particular that includes the fittest genotype here. And as you could have guessed from the previous graph, even the 16 different genotypes, this may not always be the case. So these models serve as a good approximation as long as we are near the maximum fitness of the landscape, or near a fitness peak. But trajectories on fitness landscape can be quite complex, as we can see from this diagram. That's a paper by Peter Schuster. So suppose the population starts here at the bottom in the green point, and then natural selection directs it to the maximum fitness here, which is the peak, the red point here. So then the population climbs up this mountain, namely its fitness increases, but these complex fitness landscapes can be rugged. So as you can see here, it's not smooth, there are bumps here, and sometimes the fitness drops in the way. We have valleys on the way, and then in order to climb to the top, the population needs to cross this valley. So this thing does not exist in the simple model that I showed before. Another difference, however, is that while there is a valley here in this dimension that we're looking at, and because the fitness landscape is high dimensional, then maybe there is no such valley in other dimensions. So this is a cartoon that tries to show this. So in this dimension, there is a valley, but maybe in some other dimension, not depicted here in the figure, then it's flat, there is a plane, and then the population can make a detour and avoid this valley and then keep climbing from the other side. So this seems a bit not unintuitive in the beginning, but actually in high dimensional fitness landscapes, then it's not so common to find the true fitness maxima. That is because a real maximum is one that all its single point mutations have a lower fitness. And the higher the dimension, then there are more options for single point mutations, and the probability that all of them are less fit gets lower and lower with the dimension. So then it is more likely to actually find, instead of maxima, saddle points. Okay, so after all this introduction, then we came to the question of we have these large amounts of empirical fitness landscape data. What can we learn from them? So the ambitious question that we asked in the beginning of this project was, can we take such an empirical fitness landscape data and then infer which genotype is expected to be the wild type allele? So this was our starting point, and I'll tell you what we found here. So for this project, we used an already measured and published data set of Cerevisia gene encoding for the tRNA CCU arginine. So these are measurements that were published in these two papers. They were done in the lab of Jianxi Zhang, published in 2016 and 18. So this is a figure of this tRNA molecule when it's folded. So it has 3 loops, as you can see. This is the anti-codon loop, where it attaches to the AGG codon. And on the other side, then it attaches to the arginine amino acid. Why was this gene such an attractive target for this experiment? So first, it is relatively short, only 72 nucleotides long. So having spoken about the dimensionality of landscape, so here we have a relatively small landscape of only 4 to the 72, still large. And it is also a single copy non-essential gene, namely the organism is viable even if this gene is deleted, because the AGG codon is also recognized by a wobble by another tRNA molecule, the UCU arginine. So that means that many of the mutants of this gene are still viable. So in this set of experiments, the authors created a library of the beginning 65,000 different variants of this gene, which covered all the single mutants, a large proportion of double mutants, and so on, until genotypes that were even 9 mutations from the wild type. And 23,000 of these genotypes were then measured in 4 different conditions, which include the high temperature, 37, low temperature, 23, 30, which is the best condition for growing yeast, and then oxidative stress, also in 30 degrees. So just a few words about the experiment itself. So there was a random mutagenesis where every position was mutated with a probability of 3% and equal probability to mutate to any of the 3 other nucleotides. Then a single copy of the gene was transformed in the exact location of the native gene into a strain in which the native gene was deleted. And then all these variants were grown together in a competition assay in one of the 4 conditions that I mentioned. And cells were harvested from this culture at 2 time points, time 0 and then 24 hours later, and they were sequenced. And now in the sequencing, the gene variant also served as a barcode, as an identifier. And then sequencing was used to quantify how many copies of each genetic variant are there at the beginning and at the end of the experiment. And then fitness was calculated based on this number of copies. Okay, so the formula for calculation of fitness, so fitness of genotype I was the ratio between the number of reads of the genotype at 24 hours divided by the number of reads at time 0, and that was relative to the wild type number of copies. To the power of G is the number of generations of the wild type in 24 hours. So if the cells grew at the exponential phase throughout the experiment, which is a good approximation, considering how it was done, then this is simply 2 to the power of the excess growth of genotype, excess exponential growth of genotype I relative to the wild type. So you can see already from this definition that the wild type fitness is 1 by definition here for each of the conditions. Okay, so what does the data set look like then? So since the wild type is the one that was mutated, the data set is centered around the wild type. I will use from now on the notation of N1 for single mutants, N2 for double mutants, etc. We have full coverage of the single mutants, and then it gets sparser and sparser the further away we go from the wild type. So white circles here represents genotypes that were not measured in this experiment. So we don't know their fitness. Okay, so the first thing that we looked at is look at the distribution of fitness values in all 4 conditions. And this is what I'm showing here. So distribution, and you see here every color here is a different condition. And the red vertical line here is the wild type fitness, which is 1. So quite surprisingly, we found that between 8 to 10% of the genotypes are fitter than the wild type in each of the 4 conditions. So we were a bit surprised in the beginning. So this seems not to be in line with our expectations from the mutation selection balance model that I showed before. And then we thought what could be the possible explanations for that. So first we asked perhaps this is some experimental artifact or maybe some limited accuracy of the measurements, and we thought of a few options here. So first, this experimental work was also analyzed. There was a thorough analysis of errors in the original papers, and we also repeated some of these analyses. I can say a few words about it at the end if we have time. But then the conclusion was that while there are some error sources here, that they cannot explain such a large number of high fitness genotypes. So we're talking about 2,000 different genotypes. So the second explanation was perhaps the wild type is not the fittest in any single condition, but it is optimal on average on multiple conditions. So I'll show you that this is not the case, at least for the 4 conditions that we have here measured. Third option, perhaps the wild type is a local fitness maximum and the population just stopped there. So this is not the case either. Or maybe the population simply didn't reach mutation selection balance yet. So we think this is also not very likely. Instead, what we found is that the wild type is robust to mutations. Okay, so this was just an overview, and now I'll go slowly step by step and show each of these claims and how we tested it. So the first possible explanation that we looked at is maybe the wild type is suboptimal in any single condition, but it is the fittest on average. So that would imply that the genotypes that seem to be fitter in one condition should be much less fit in other conditions. Okay, so what we did here is simply look at the correlation and fitness values between conditions. So we plot here the fitness of genotypes in 3 conditions, 23, oxidative stress, and 37 degrees, against their fitness at the 4th condition, which is 30 degrees. So the left graph is for the high fitness genotypes. The right one is for the low fitness genotypes. So you can already appreciate that for the high fitness genotypes, there is a quite good correlation, which means that the genotypes that are very fit in one condition are usually very fit in all 4 of them. This is not, by the way, the case for the low fitness genotypes. There the correlation is much lower, but still it is rare to find that a genotype that has low fitness in one condition would be very fit in some other condition. Okay, so this does not seem to be the case. However, I must say that we have here data only of 4 conditions. So while they are considered typical growth conditions for yeast, we still cannot rule out the possibility that in nature there could be additional conditions in which the wild type behaves very differently and may be optimal there. Okay, so another way to test that was to define the geometric mean fitness of these genotypes. So if we assume that all 4 conditions are equally likely, and if we assume that the switching time between conditions is much shorter than the typical time between mutations, the evolutionary time, then we can consider simply the geometric mean fitness over all 4 conditions. So we calculated it, and here we have the distribution of the geometric mean, and the red here is the wild type. So now it's not 1 because we had to rescale the values relative to one of the 4 conditions. So still in geometric mean fitness, we have a proportion of genotypes that have a higher geometric mean fitness. We said, okay, maybe we should look, we should be more harsh, and maybe the conditions are not equally likely. So we even looked at the ranking of the genotypes, and here we show the minimal rank of fitness rank of a genotype. So high rank here is a high fitness, low rank is low fitness. And this is the minimal rank and the average rank of the wild type. And still there is a proportion of genotypes with higher minimal rank, namely they are fitter in all 4 conditions, and same for the average. Okay, so this seems to rule out this explanation, at least for the 4 conditions that we have data for. Okay, so then we went on to the next explanation. So maybe the wild type is a local maximum. But before that, we asked where exactly are these fitter than wild type genotypes. So here we dissected the genotypes according to the number of mutations from the wild type. So we have here simply fitness. This is for geometric mean fitness here. Geometric mean fitness of the single mutants in red, double mutants in green, in blue, and triple and quadruple mutants. And this is the wild type here, the black line here. So we see that we have fitter than wild type genotypes in all 4 genetic, I call them genetic neighborhoods. So even among the single mutants, there are fitter genotypes. However, most of the fitter than wild type genotypes are double mutants. They are in N2, the blue one. Okay, so to put numbers here, so in N1, the single mutants, we found 4 different genotypes. And in N2, the double mutants, we found 730 different genotypes that are 9% of those that were measured, which have a geometric mean fitness larger than that of the wild type plus some delta. So we took this delta as a margin just because of the experimental error. Okay, so we can already tell that the wild type is not a local maximum because we found even single mutants that are fitter. But we wanted to know more than that. So what about the many double mutants that are fitter? So are they accessible from the wild type? So then we looked at that too. We simply mapped all the evolutionary trajectories leading from the wild type to the fitter double and triple mutants. And then we asked how many such trajectories are such that fitness increases in every single step of them. Okay, so we go up here, 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 and here. Same here. So we did this, I'm showing here the numbers for the geometric mean fitness. We also did this for the fitness data in a single condition. There we got even higher numbers. So for the geometric mean fitness, so if we require that at every step the fitness increases by at least 1%, then we find the 771 trajectories of length 2, namely leading to fitter double mutants. If we are stricter and require that the fitness increases by at least 2% in every step, then we still find 159 such trajectories. So we went on to the triple mutant. So even there we find 277 or 26 trajectories, depending on which fitness increase we require at every step. So the message here is that we find a large number of such trajectories in which fitness increases. We're not talking about just one or two possibilities. And I would say even more than that. So this is an underestimate of the actual number of trajectories, because we only included here trajectories that were fully in our data set. So if there could be another genotype that was not measured, we wouldn't count trajectory passing there. So I would guess that there are additional trajectories that were not included just because these genotypes were not measured. And of course, trajectories can also pass through a neutral transition. So a minimal fitness increase is not necessary. So we find trajectories from the wild type to fitter genotypes between 1 to 3 mutations away, and quite many of them. So why then is this genotype the wild type? Or in other words, what is then unique about the wild type? So I'll go back to the previous slide, and now let's look at another interesting thing that we can see from it. So this is the fitness distribution for the single, double, triple, etc. Mutants. So if you look closely, then the distribution of the single mutants, the red one, is much narrower compared to the double mutants, for example, the blue one, and also to the other two. So that means that the distribution of single mutants, that the single mutants have fitness values that are relatively close to that of the wild type. So this seems interesting, and we asked ourselves, is this typical? Is it common to other genotypes in the landscape? Perhaps the landscape is simply very correlated. Then we came to think that perhaps the wild type is more robust to mutations compared to other genotypes, because it looks like its single mutants do not differ by much from it. So to test the last thing of robustness to mutations, we then had to define some measure of what is the sensitivity of a genotype to mutations. So we defined the following measure. So I'll go here step by step to explain this. We call it mutational fragility. It accounts for the genotype sensitivity to deleterious mutations only, and we do not consider beneficial mutations here. So suppose that this is the genotype we're interested in, and those in the circle are the single mutants, and color represents fitness here. And the green or blue is fitter genotypes, and yellow and red are less fit genotypes. So then this measure only considers the red and yellow ones. It is simply the average of the fitness difference between the genotypes. So our focal genotype is I, and the J's are all the single mutants. And we take this only if Fi is bigger than Fj, namely this is a deleterious mutant. And otherwise, if Fj is larger than Fi, this is a beneficial mutant, then we take here 0. Okay, so this is the definition of the measure. Which genotypes are expected to have a low or high fragility? So a genotype that is very fit here on the top is expected to have high fragility because the single mutants here are deleterious. A genotype that is on a plateau or one that has very low fitness are both expected to have a low fragility, because here the single mutants are similar in their value, and here the single mutants are beneficial. So then they would be accounted here as a 0. So to get an intuition of how this measure behaves, we simulated a landscape using the NK model. This is the distribution of the fitness values here. And here we plot the fragility against fitness of all the genotypes in this simulated landscape. And indeed, we see that for the low fitness genotypes, there is a low fragility. Their single mutants are beneficial. And somewhere in the midpoint, it starts to rise. And the most fragile ones are those that have a high fitness, as we expected. Okay, so how does it look like in our data set then? So this is the analogous plot of the mutational fragility against fitness for the tRNA data set. So the black line, the black circle here is the, the wild type. and then the single mutants are the red ones, the double mutants are the blue one, and there are a few triple mutants, they are hidden here. Okay, so we see here something quite interesting. So the wild type is here at the tip of the cloud. It shows some balance between fitness and fragility. So it is amongst the least fragile genotypes in the data set, but amongst the low fragility, but not the low, really the lowest one, but amongst relatively low fragility, it is one of the fittest genotypes. And then looking at this axis, then if we look at genotypes that have similar fitness value to the wild type, then amongst them, it is one of the least fragile. Also, if we compare it to its single mutants, then as we saw before, many of them have similar fitness. So they are concentrated here near 1 fitness. So amongst them, it is one of the least fragile genotypes. Okay, so to answer the previous question, then what we see in we saw for the wild type seems not to be typical. So the wild type is unique in where it is located in this space. So just a few words about the technicalities here. So there were lots of technical challenges just because we were working with an incomplete data set that had many holes, and the definition of fragility was based on having all the single mutants of a genotype. And actually we had all the single mutants only for the wild type. And for most other genotypes, we had much smaller numbers of their single mutants. But not only that, the problem was even worse because the data set was created by mutagenesis of the wild type. So it was biased to have almost all the genotypes when we get close to the wild type, and fewer the further away. So just to demonstrate this. So let's think that this is our genotype and these are all its single mutants. Here is the wild type, and the light blue are closer to wild type. The darker blue have equal distance to wild type, like this genotype, and yellow ones are further away. So just because of the way the dataset was created, we usually have all or almost all of the blue ones, those that are at equal distance or closer to wild type, and we have only a few of the yellow ones, those that are further away. But actually the yellow ones are the majority. So we have a biased representation of the nearest neighbors for most of the genotypes in the data set. And then we had to correct for that in this calculation. So how did we do this? So we divided the neighbors data set into 3 subsets, as I showed here. So those that are closer, equal distance, or further away from the wild type. We calculated the contribution to this measure for each of these 3 subsets separately, and then we weigh them according to the true size of the set that they represent. So this was mostly about the further from wild type genotypes that received a much larger weight than their number here. So this is how we corrected for this bias in the data set, and the data that I showed before represents that already. Okay, so a few words about errors here. So I mentioned that at the beginning. We analyzed here two possible error sources. So one of them was error due to a read count noise, namely the fitness estimate is based on the number of reads, and this is only a single sample, and it could create errors here. And another thing is simply biological variation. So there were biological repeats for the experiment, and the actual reported fitness values were the average over multiple biological repeats. So what we did here is taking the raw data of the number of reads for the distinct repeats. And then we assume that this is a binomial random variable, and we sampled from this distribution multiple times. And we use these samples to calculate, to estimate actually the distribution of fitness values that we could have received here. So each color here represents the estimated fitness distribution for a single biological repeat of the experiment. This is for a particular genotype that I chose here. So you can see here that at least in this case, then the read count distribution is relatively narrow, and what seems to be the dominant source of noise here is the difference between biological repeats. So in some of the cases, it's not that great. It sometimes overlaps, but still the biological variation seems to be amongst the largest sources. So what I'm showing here is the calculation of the standard error of the mean over the multiple repeats of the experiment against the mean value here. So we see here that for almost all of the experiments, all of the genotypes, then it's below 5%. And for most of them, it's even below 2%. So that's the accuracy of that we can relate to the fitness values that we have here. Okay, so we saw here that the wild type seems to be robust to mutations. And we went back to the literature, and the robust or so-called flat genotypes were demonstrated in very few cases in the past. So this was done in digital organisms, in computer simulations, in the famous paper by Klaus Wilke, the survival of the flattest. And then experimentally in viruses, RNA viruses. But what is important is that these organisms in which flat genotypes were demonstrated have very high mutation rates. So we're talking about 10 to the minus 4 to 10 to the minus 5 per nucleotide per replication. And the gene that we analyzed is from yeast that has a much lower mutation rate. So it's around 10 to the minus 9 here. So although tRNA genes were reported recently to have a higher mutation rates by 10-fold compared to the background genome, then still we're talking about a lower mutation rate compared to the viral one. So then this still leaves open the question of what is the underlying evolutionary mechanism here, and did actually the tRNA gene evolve to be mutationally robust, and this is the reason for the wild type allele that is suboptimal but still robust to mutations that we see here. So I don't have a closed answer for that, so this is still left here as a mystery, but I'll be happy to discuss with people if somebody has ideas here. And to summarize, so what I've shown you here is that the yeast tRNA wild type allele, the tRNA CCU arginine, is suboptimal. It is not the fittest on average on the 4 conditions measured here. It is not a local maximum either. It is, however, the least sensitive to mutations compared to single mutants and other genotypes with a similar fitness. The evolutionary mechanism that led to this fitness robustness balance still remains unknown. And also with the growing number of mass fitness measurements, then appropriate analysis method should be developed, like the thing that I've shown you here with the fragility and how to compensate for the incompleteness of the data set. So this was a joint work with my collaborator, Tzachi Pilpel from the Weizmann Institute, and most of the work that you saw here was done by our joint student, Tzachi Gabzi. And this is it. Thank you for listening, and I'll be happy to take questions.